microbiology. So, okay, for number one, no, so who introduced the importance of hand washing and considered as the father of hand hygiene? So this particular person, no, so advocated hand washing during surgeries. No? So for number one, the best answer here would be letter C, Semmelweis. Okay, so this would be letter C, Semmelweis. No? So for the other choices, no, Lister, so is the one who introduced the use of phenol as an antiseptic. So Robert Koch, so advocated germ theory, and Paul Ehrlich, no, so contributed the use of salvarsan for treatment of syphilis. So he's also the one who introduced the concept of the side chain theory. So side chain theory in antibody production. Okay, so for number one, this is letter C. Okay, for number two, no, which of the following is incorrect? So keyword natin dito, so incorrect. So it is incorrect regarding the cell wall, the bacterial cell wall. The best answer here for number two is letter B. Okay, so it is rich in sterols that provide strength and stability. So you have to take note, no, sterols are not present. They are not present in bacteria. So except in mycoplasma and also ureaplasma. So these two groups of bacteria can contain sterols present in the cell membrane. Okay, so you don't find sterols in the cell wall. Okay, so you don't find this in bacteria, no, except in mycoplasma and ureaplasma. Okay, so in mycoplasma and ureaplasma, you can find the sterols in their cell membrane. Okay, number two, this is letter B. Number three, so you are assigned in the microbiology section and tasked to demonstrate endospores. So endospores, no, these are highly resistant structures that are found inside bacterial cells, particularly spore-forming bacteria such as your Bacillus and Clostridium. Okay, so these are gram-positive bacilli that are capable of producing spores. Okay, so endospores, the tackhead bacillus. So this particular organism in tackhead bacillus, that's your Clostridium tetani. Okay, so what can you use to demonstrate your spores? So definitely that's not your gram stain. So the gram stain cannot stain the endospores. No, so if you do a gram stain of bacillus or Clostridium, the endospores will just appear colorless. Okay. Amphidian is also not the answer since that's a stain for the capsule. That is a capsular stain for anthrax, for Bacillus anthracis. Lamb, so that's your methylene blue, Loeffler's alkaline methylene blue. So we use that for the demonstration of the metachromatic granules. So we encountered the metachromatic granules in Corynebacterium. And Jimenez is also not the answer. We use that to demonstrate or to stain Rickettsia. So for number three, the best answer here would be letter B, your Schaefer-Fulton. For the Schaefer-Fulton, okay, the Schaefer-Fulton makes use of your malachite green to stain the spores. Okay, that would be used to stain the spores color green. And you have also your safranin. Safranin would stain your bacterial cell. Okay, your bacterial cell, it would be color red. Okay, using safranin. So for number three, this is letter B. Okay, number four, which of the following bacteria are not usually demonstrated using gram stain? So although your gram stain is the fundamental staining technique that we use in microbiology, so your gram stain cannot be applicable to all types of bacteria, no, especially those bacteria that do not have a cell wall, as well as those bacteria that are found intracellularly. Okay, so among the choices for number four, the best answer here would be letter E. So both B and C. So chlamydia and mycoplasma are usually not gram stained since chlamydia is intracellular. Mycoplasma do not have a cell wall. So instead of gram staining, we can use your fluorescent stains to demonstrate them. Okay. So for number four, this is letter E. Okay. Number five, no, not a function of the capsule. So which is not a function of the capsule. So the capsule, this is a structure that surrounds the bacterial cell. Okay. So this can be made up of polysaccharides. Okay, primarily polysaccharides. Okay, or even glutamic acid, no, depending on the type. But primarily they're usually made up of polysaccharides. Okay. So which is not a function of the capsule. So the capsule. Okay. So the answer here for number five is letter C. So it does not give the shape of the bacterial cell. So it is the cell wall that gives the shape. Okay, so functions of the capsule include it's antiphagocytic, it prevents drying or desiccation. Okay, it's also antigenic, but it would not give the shape of the bacterial cell. Okay, so examples of bacteria that are capsulated include your Klebsiella pneumoniae, Streptococcus pneumoniae. Okay, so for number five, this is letter C. Okay, number five, letter C. Number six, which of the following instruments or apparatus is not needed in preparing your plated media? Okay, so in the laboratory, so we are also preparing different types of culture media. So the culture media can be dispensed in tube or in plated or in your Petri dishes. Okay, so you need to properly measure the amount of water. So you use a graduated cylinder for that. No? Okay, so to measure the amount of powdered media, you need also your analytical balance. But which is not used in preparing plated media. So that is actually your letter B, your beaker. The beaker is primarily if you are for preparing tube media. Okay, so if the media is to be dispensed in test tubes, now we dissolve the powdered media in a beaker for the tube media. But if you're preparing plated media, you dissolve it, no? You dissolve it in the Erlenmeyer flask. Okay, number six, this is letter B. Number seven, which is a culture media that is selective and differential. No? So if it's a selective culture media, it has inhibitors. So to prevent the growth of other bacteria. No? So if it's differential, it has a pH indicator. So it can distinguish, no, whether or not the colonies are able to ferment a particular sugar or not. So there will be color change. No? So for number seven, the best answer here. Okay, the best answer here, no, which of the following is a selective and differential media, that is letter B. Number seven, letter B. That is your mannitol salt agar. Mannitol salt agar, this is selective and differential for the members of Staphylococcus. Okay, so primarily, it would only allow the growth of staph. So it has inhibitors, no? So it has a high amount of salt to inhibit other bacteria. So around 7.5%. And it has a pH indicator. pH indicator is phenol red. So since it is a differential medium, no, it has a sugar. That is mannitol. No? So to determine if it's able to ferment mannitol or not. Okay. Number seven, letter B. So blood agar plate, that is enriched and differential. Enriched and differential. BSA is just selective for Salmonella typhi. TSI is a biochemical medium. SEA is also a biochemical medium. Okay. So number seven, this is letter B. Okay. Number eight, mismatch, which is the following is mismatch. So we have the following culture media and their functions or uses. No? So for number eight, what is mismatch here is letter E. So PEA. PEA is your phenyl ethyl alcohol agar. So phenylethyl alcohol, agar, is just a selective media. Okay, it's just selective, no? So it is not differential. So it is selective for gram-positive cocci, but it's not differential. Okay, so the other choices are correct, no? So TCBS for Vibrio, Tinsdale for Corini, CCFA, that's useful for Clostridium difficile, LJ, that's your Lowenstein-Jensen, which we can use for mycobacteria. Okay, for number 8, this is letter E. Okay, number 9, what's the expected appearance of colonies if you have Salmonella enteritis isolated on XLD? XLD, that is your xylose, lysine, desoxycolate or deoxycolate agar. Okay, so this is a highly selective and differential media for the growth of enteric bacteria such as Salmonella and Shigella. Okay, so very selective for Salmonella as well as for Shigella. No, so XLD also has an H2S indicator. So if it is an H2S producer, you have a black center. Okay, for number 9, the best answer here would be if you're dealing with Salmonella. So the best answer here would be letter B, red colonies with a black center. Okay, black centers in Salmonella is H2S positive. Okay, it's red colonies. No, although your Salmonella can ferment xylose. Okay, but because Salmonella is LDC positive and XLD has lysine. Okay, so there can be the production of LDC, okay, lysine decarboxylase, no, that would produce cadaverine. Okay, cadaverine is alkaline, so making the colonies color red. Okay, so number 9, this is letter B, no, red colonies with black center. Number 10, so what is the predominant indigenous flora of the colon? So what is in other words, the normal flora of the colon or the large intestine? So primarily for the large intestine, so you have a large amount of bacteria there, particularly normal flora, and they're usually anaerobic bacteria. Okay, so particularly this would include your gram-negative anaerobic bacteria. So the best answer here would be letter D. So one of the predominant anaerobic bacteria present in your colon would include your Bacteroides fragilis. Okay, so that is an anaerobic gram-negative non-spore forming bacteria. Okay, so predominantly you have letter D. Okay, for number 10, so that's letter D. Okay, number 11, so what's the difference between aerobes and microaerophilic organisms? So these two groups of bacteria or organisms would differ with regards to their oxygen requirement. Okay, for number 11, this is letter B, so we are talking about their oxygen requirements. Okay, so aerobic organisms would prefer growing in the presence of oxygen, so approximately around 18 to 21% of oxygen. Okay, on the other hand, if it's microaerophilic, they need to have a reduced amount of oxygen. Okay, so that would be around 5 to 10%. Okay, so microaerophilic organisms include your Helicobacter as well as your Campylobacter. Okay, so these groups of bacteria, your microaerophiles or microaerophilic, would require reduced amounts of oxygen. Okay, number 11, this is letter B. Okay, number 12, which is a gram-positive bacilli. So which of the following is a gram-positive bacilli? The best answer here for number 12 is letter B, Corynebacterium. Okay, although Staphylococcus is gram-positive, okay, Staph is gram-positive cocci. Okay, Corynebacterium is the best answer since this is a gram-positive bacilli. Okay, so these are non-branching bacilli, and they're also catalase positive. Okay, Corynebacterium. No? Veillonella, these are anaerobic gram-negative cocci. Vibrio is gram-negative bacilli. Okay, Bordetella is also gram-negative. Okay, for number 12, this is letter B, Corynebacterium. Okay, number 13, so questions on milk bacteriology. So an agent of red milk that can be isolated in milk. No? So milk is considered sterile. Okay, so the milk coming from cows is actually considered sterile, but the milk collected from the cows can be contaminated. No? They can be contaminated. Okay, so because of the contact of the milk with the skin of the cow or from the water or the apparatus or utensils used in collecting the sample. Okay, so microorganisms that can be present in the milk can either be pathogenic or non-pathogenic. Okay, so they can be pathogenic or non-pathogenic. Okay, so the agent of red milk is okay, so the agent of red milk here, the best answer here would be letter P, Serratia, no? Serratia marcescens. So Serratia marcescens, if you recall, that has the prodigiosin pigment, giving that red color. Okay, so Pseudomonas for the other choices, like your Pseudomonas cincinnea. So cincinnea, it's chromogenic, so it would usually give that blue, okay, blue color milk or blue milk. Okay, Subtilis, okay, so it usually has a proteolytic action on milk. Okay, Strep lactis is responsible for the souring of milk. Okay, and Aeruginosa is also chromogenic. It would usually give that blue-green milk. Okay, blue-green milk. For number 13, the best answer here would be letter B. Okay, number 14. So which of the following can exhibit hemolysis on human blood? So meaning, so this particular bacteria can hemolyze blood, no, and can grow on HBT. So that's your human blood bilayer twin agar. So which bacteria can grow in HBT and is hemolytic in human blood? This is your Gardnerella vaginalis. So Gardnerella vaginalis, okay, can cause bacterial vaginosis. So it can be sexually transmitted, no? Your bacterial vaginosis, no? So females can have that discharge, no? And the presence of flu cells, okay? Presence of flu cells, no? In patients infected with this organism. Okay, so Gardnerella vaginalis is gram-positive. It is a gram-positive bacilli that is catalase negative. It's catalase negative. It's also non-motile. Okay, for number 14, so this is letter A. Okay, letter A. Okay, number 15. So what would be the most appropriate specimen in diagnosing food poisoning caused by Bacillus cereus? No? So cereus, that is an agent of fried rice poisoning. Okay, fried rice poisoning. So this is a spore-forming bacteria, no, implicated in fried rice poisoning. Okay, and patients with food poisoning caused by Bacillus cereus can either have the emetic form or the diarrheal form. Patients with the emetic form would usually complain or experience vomiting. Okay, so one of the pronounced or predominant manifestations in emetic form is vomiting. No? If it's diarrheal, of course, you have gastrointestinal symptoms. Okay, so diarrheal. So if you're dealing with food poisoning, the preferred sample, of course, is the letter A, the implicated food. Okay, so that would be the general rule regarding food poisoning. So it's best to submit or collect the food sample. Okay, number 15, letter A. Number 16. So what is the most common agent of meningitis among adults? So meningitis, so that is the inflammation of the covering of the brain or the meninges, no? And this can be caused by bacteria, viruses, fungi, no? And even parasites, no? So but among adults, the most common of bacterial meningitis. Okay, bacterial meningitis is of course, no, for number 16, this is letter C, Streptococcus pneumoniae. So strep pneumo is still considered as the most common agent of meningitis among adults. So aside from meningitis, strep pneumo is also implicated in community acquired pneumonia. Strep pneumo is also important in causing ear infections or what we call as otitis media among children. Okay, among children. Okay. So for the other choices, these are also agents of meningitis, but for strep agalactiae and even your listeria, they can cause meningitis usually among neonates. Mycoplasma pneumoniae is known for causing the atypical pneumonia, or what we call as the walking pneumonia. So walking in the sense that patients who have this type of pneumonia are still able to work, okay, and it is a less severe type of pneumonia, okay, if it's mycoplasma pneumoniae. Okay, so for number 16, the best answer here is letter C, strep pneumo. Okay, for number 17, so fried egg colonies stained with DNS. So what is that particular group of bacteria that has the fried egg colonies? Okay, that can be stained by DNS. So DNS is basically composed of methylene blue. So the addition of the DNS stain would make the colonies, the fried egg colonies, okay, easily seen. No? So fried egg colonies, of course, are typical of letter C, Mycoplasma pneumoniae. Okay? So mycoplasma would have the fried egg colonies. No? So mycoplasma produces very small colonies. No? So in fact, no, mycoplasma is considered as the smallest culturable bacteria. Okay? So smallest culturable bacteria. Okay? And they would have that fried egg appearance. Okay? Fried egg appearance. So for the other choices, no, so such as your actinomyces, Israeli, they have that molar tooth colonies. Moraxella would have that hockey puck consistency of the colonies. Yersinia would have that hammered copper appearance of the colonies. Okay, for number 17, this is letter C. Okay, number 18, how can we differentiate staph and strep? So what test particularly can differentiate these two gram-positive cocci? So both staph and strep are gram-positive. Okay, gram-positive cocci, so definitely we cannot use your gram stain. Okay, so in order for us to distinguish the two, it's best to perform your catalase test. Okay, so catalase test makes use of 3% hydrogen peroxide. Okay, so staph would be positive for catalase, strep would be negative for catalase. So the positive result in the catalase test is bubbling or effervescence. Okay, number 18, letter A. Number 19, which is not an appropriate, okay, keyword not appropriate or not useful, anticoagulant for culture. So for number 19, the best answer here would be letter D, EDTA. Okay, so EDTA, although it's an anticoagulant, EDTA is not for culture. Okay, we don't use that if you are planning to culture or cultivate the organism. No, so EDTA can be used for molecular testing. Okay, for molecular testing, nucleic acid amplification tests, EDTA can be used. Okay, for the other choices, we can use that, no, such as SPS, which is the commonly used anticoagulant. No, okay, for blood cultures. So usually this is for blood cultures. Citrate can also be used, no, as well as heparin. Heparin can be useful for viral cultures in the blood. Okay, but definitely number 19, not EDTA. Okay, so the answer here, not for culture, is letter D, EDTA. Okay, number 19, letter D. Number 20, which is incorrect. Paired statements is incorrect. No? So we have here the organism and their associated disease or syndrome. So for number 20, so which is incorrect here, this is letter C, strep agalactiae and acute glomerulonephritis. Okay, so this is incorrect since we don't associate agalactiae with glomerulonephritis. Agalactiae are usually associated with meningitis, sepsis, no? So among infected neonates, okay, but not glomerulonephritis. If it's glomerulonephritis, we usually associate this with the sequelae of Streptococcus pyogenes. Okay, so glomerulonephritis. Okay. The other choices are correct. No? So staph aureus, that's associated with your Ritter-Lyall disease, okay, or your scalded skin syndrome. For meningitis, so that's your Waterhouse-Friderichsen, so affecting the adrenal glands. And for gonorrhea, if it affects, okay, the covering of the liver of women, so that's your Fitzhugh-Curtis syndrome. Okay. So for number 20, so the best answer here is strep agalactiae. Okay, and acute glomerulonephritis. Number 20, letter C. Number 21, also highly associated with MRSA. What particular gene? So MRSA, that's your methicillin-resistant staph aureus. No? So the gene, no, responsible for that, and the detection of this gene is also considered as the gold standard, okay, in the detection of MRSA. So the gene that we associate with MRSA is letter A, your MECA. Your MECA gene. So number 21, letter A, MECA gene. So that is the gene that so that would be associated with MRSA. So the MECA gene, okay, codes for your PBP2A. So this is an altered protein of your staph aureus, no? So leading to methicillin resistance. Okay. So the other choices like ERM, that's usually associated with the D-zone test. Okay, tox gene, that's for the diphtheria toxin. VAN-A, that's usually associated with vancomycin resistance. Okay. Number 21, letter A. Number 22, what is the standard inoculum for the disc diffusion method? So for disc diffusion, that's your Kirby-Bauer. So definitely for antibiotic susceptibility testing, you need a standard inoculum for the susceptibility testing. Okay. And the standard inoculum, okay, of your Kirby-Bauer or the disc diffusion method, okay, should be compared to the 0.5 McFarland standard. Okay. So to the turbidity of the McFarland standard, 0.5. Okay. So if it is the same turbidity with the 0.5 McFarland standard, we are assured that the inoculum contains, okay, letter A, 1.5 times 10 to the 8 CFU per ml. So if the turbidity of the inoculum is the same with the 0.5 McFarland, so the inoculum is already standardized and it contains 1.5 times 10 to the 8. Okay, number 22, letter A. Number 23, so you have here the Medtech working on a wound culture, aerobic wound culture. So the colonies appear large, gray, non-hemolytic or gamma hemolytic. Okay, so, and when lifted using the inoculating loop, the colonies has that consistency of whisk egg whites. Whisk egg whites or tenacious consistency. No? So gram stain reveals a gram-positive bacilli. What is the most likely identification of this colony? Okay. So since this is an aerobic organism and it's gram-positive, we can already remove Francisella and Burkholderia. These two are gram-negative. Okay. So Nocardia asteroides, okay, so are also not included since these are usually branching. branching bacilli. Okay, and they're more dry. The colonies appear more dry, and they're also partially acid fast. Okay, so since it's a wound culture, the best answer here would be letter A, anthrax, no? Bacillus anthracis. So what are the important keywords in anthrax? It's non-hemolytic. Okay, you have that medusa head colonies, non-motile, and you have the tenacious or egg white consistency. Okay, number 23, letter A. Okay, Cereus is beta hemolytic, by the way. Okay, so number 23, letter A. Number 24, in a broad dilution setup, the test antibiotic is diluted fourfold. For the first statement, so in your dilution susceptibility testing, we of course dilute the antibiotic, no, but not fourfold. Okay, it should be twofold dilution. Okay, so the first statement is wrong. The second statement, the MIC is the lowest concentration of the antibiotic, showing no visible growth. So if we are determining the MIC, no, so we determine the lowest concentration of the antibiotic, showing no visible growth or no turbidity. So this is a true statement. Okay, so for number 24, this is letter B. Okay, number 24, letter B. 25. So which of the following susceptibility methods does not provide an MIC value? So definitely, so this is your letter B, your Kirby-Bauer method. So that's your distribution. So in your distribution method or Kirby-Bauer, now we are measuring here zone of inhibition. Okay, so expressed in millimeters. No? So based on the zone of inhibition, no, we can now then classify whether it's susceptible or resistant. No? Okay, the other choices would all provide you an MIC value, such as your E-test or epsilometer test. Okay, you have also the Brock dilution and agar dilution. Okay, but not your Kirby. Number 25, letter B. Okay, so number 26, no, you have an intern working in the lab, no, and reported the different results. No? Which of the following results here on the choices is an urgent alert value? Okay, so meaning if it's an urgent alert value, this is a panic value. So meaning the result should be reported immediately. It should be reported immediately to the attending physician or doctor. So among the choices, now, which is a panic value that should be reported immediately. So this is letter A. So wound culture containing strep pyogenes. So why is this a panic value? No? You have to take note of the presence of strep pyogenes in tissue or in wound culture can indicate necrotizing fasciitis. So this may be a case of necrotizing fasciitis or flesh-eating bacteria. Okay, so that has to be reported immediately. Okay, the other choices, so there might be some delay. You may allow some delay in the reporting, so it's not a very urgent value. No, so presence of ascaris eggs, no? Or urine culture due to E. coli and sputum showing gram-positive cocci. No? So gram-positive cocci can be seen normally in your sputum. Okay, but wound culture with strep pyogenes, that is definitely a panic value. 26, letter A. 27. So as a general rule, no, specimens for microbial culture should be processed within 2 hours. Okay, so this is letter C. So 2 hours. Now, so if there's any delay in the processing, so for more than 2 hours, we can put the specimen in the refrigerator for a short period of time, or we can place the sample in transport media to maintain the viability. Okay. For number 27, this is letter C. Okay. Number 28, in a conventional gram staining technique or procedure, how long should we flood the smear with grams iodine? Grams iodine, that is the mordant of your gram staining technique. So for your grams iodine, so that is 1 minute. Okay. Number 28, so grams iodine should be flooded to your bacterial smear, no, for 1 minute. Number 28, letter C. Okay, number 29. What do you think is the function of phenol in your conventional acid fast staining technique? So for the conventional acid fast staining technique, so this is your Ziehl-Neelsen or the hot method, or the Quignon method, which is the cold method. So the phenol, or this is your carbolic acid, okay, carbolic acid, this functions as the chemical mordant and solvent. Okay, number 29, letter B. So it's a mordant, it's a chemical mordant. Okay, so it can also function as a solvent since this is added added in the carbolfuchsine, to dissolve the fuchsine dye. Okay, so that's used to dissolve the fuchsine dye in the carbolfuchsine. Okay, number 29, letter B. Okay, the colorizer in acid fast, that's your acid alcohol. The counter stain is your methylene blue. Your primary stain in your acid fast is carbolfuchsine. Okay, number 29, letter B. Number 30, so which is an agent of culture negative UTIs. So these are bacteria that can cause urinary tract infections, wherein your patients can experience urethritis or difficulty in urination. But in most cases, no, they would be unable to be cultured in traditional or routine urine cultures. No, so the patient experiences signs of UTIs, but usually you have a negative urine culture. Okay, so these culture negative UTIs are also usually transmitted sexually. No? So for number 30, so this is a letter C, chlamydia. Okay? Chlamydia is an example of a culture negative UTI. Okay, so chlamydia, okay, is also a sexually transmitted infection, the most common STD or STI in the US. Okay, so 30, letter C. The other choices are usually able to be grown or cultured in your routine urine CS. Okay, number 30, letter C. 31, which of the following is employed for enumeration of urinary pathogens? No? So this particular culture media is now already becoming commonly used in the laboratories. No? So for urine culture, no? So we can perform colony count in this culture medium. Okay, so this culture medium can allow the growth of different pathogens found in the urine, whether gram positive or gram negative. It's also able to suppress the swarming of Proteus. So for colony counting, so instead of using your traditional blood agar plate, so we can do the colony count in your letter C, CLED. Okay, CLED is a commercially available culture media, so which means this is your cysteine, okay, lactose, electrolyte, deficient agar. So this would allow the enumeration, isolation of different urinary pathogens, and we can perform the colony count here. Okay, McConkey is just selective and differential. HEA, we don't usually use HEA for urine culture, that's for stool culture. Okay, PEA, that's for gram positive. TCBS, if you're isolating Vibrio. Okay, so for the best answer for 31, this is letter C. Okay, 32. Which of the following might lead one to suspect that the organism is a non-fermenter? So if it's a non-fermenter, so it will not break down your sugar, in the absence of oxygen. It's a non-fermenter. So usually, if it's a non-fermenter, they are colorless on McConkey agar, and on your TSI tube, you have a K over K reaction, alkaline slant over alkaline bond. Okay. So for number 32, no? So this is letter C. Okay, so pink column is a Macankey that would tell you that's a fermenter. Okay, usually non-fermenters are oxidase positive, not negative. Okay, so the best answer here, if it's a non-fermenter, so you check the TSI, the result of the fermentation of sugars on TSI. If it's a red over red or alkaline slant over alkaline bot, that's most likely a non-fermenter. Number 32, letter C. Number 33, what's the drug of choice for MRSA infection? So we can give vancomycin. Okay, so vancomycin. Okay, so we cannot anymore give penicillins, ampicillin, since these are all related to methicillins, so they're already resistant for that. Metronidazole, that's primarily for anaerobic bacteria. It's for anaerobes, no? Cefoxitin, this is used for screening of the presence of MRSA. Okay, so number 33, this is letter A. 34, force stable odor. So colonies or cultures that has a force stable or barnyard odor. Okay, barnyard odor. So definitely, so this particular organism is known for being a spore-forming bacilli, no? Anaerobic. So this is, okay, your Clostridioides, no? Letter C, or your Clostridium difficile. So this is an anaerobic, gram-positive spore-forming bacilli that is associated with Pseudomembranous, okay, Pseudomembranous colitis. Okay, so causing severe diarrhea among elderly patients, hospitalized patients, no, who had a history of long-term use of broad-spectrum antibiotics. Okay, so cultures of C. difficile plus U. difficile would have that barnyard or force stable odor. 34, letter C. Okay, 35, the TSI result or the triple sugar iron result is yellow over yellow. Okay, so if it's yellow over yellow, so there is acid production. So acid over acid. So in symbols, that's A over A. And aside from being yellow over yellow, it also has cracks or spaces. So that is A over AG, but no blackening. Okay, so this would indicate that if you have an A over A with A over AG reaction on TSI, so this would indicate that it fermented sugars. No, not just one sugar, but more than one. No? Usually two sugars. Okay, so the best answer here, no, is letter A. It fermented glucose, okay, as well as lactose or even sucrose. Okay, so if the yellowing is only just limited to the bot, so for example, a K over A reaction, if it's a K over A reaction, it only fermented sugar, your glucose only. Glucose fermented only, no? It only fermented one sugar, no? If it's K over A. If it's A over A, so the best answer there would be letter A. 35, letter A. 36, not consistent with Staph epidermidis, no? So which of the following is not consistent with Staphylococcus epidermidis? So this is a gram-positive cocci in clusters that can be part of the normal flora of the skin, okay, but it may be associated with stitch abscess. It may also cause opportunistic infections. Okay, so although it's part of the normal flora of the skin. Okay, so which is not consistent with Staph epidermidis. For number 36, this is letter C. DNA is positive. Okay, Staph epidermidis is not DNA is positive. That is Staph aureus. Okay, so 36, this is letter C. Okay, so epidermidis is coagulase negative. That's correct. Since this is Staph, Staphylococcus, it's also catalase positive, and it's non-hemolytic or gamma hemolytic. Okay, but not consistent with Staph epi is letter C. Number 36, letter C. Number 37. So current versions of the BACTEC. Okay, so BACTEC that is considered as the first, no? So the BACTEC system is considered as the first automated blood culture system. Okay. So the current versions of the BACTEC for blood culture, no, okay, are able to detect growth in your blood culture bottles by measuring carbon dioxide. Okay, so one way of telling that there is growth in your culture is if your organisms are producing carbon dioxide. Okay, so first there are different ways to detect the carbon dioxide inside the blood culture bottle. So for the current versions of the BACTEC, okay, so they detect the carbon dioxide production, okay, by using fluorometry. Okay, so for 37, this is letter D. Fluorometric changes from the production of CO2. Okay, so the current versions of BACTEC include your BACTEC 9240, okay, 9050. So these are some examples. Okay, so the old versions or old models of BACTEC in the past, a long time ago, they used radioactive radioisotopes for measuring. But nowadays, we use fluorometric changes for BACTEC. Number 37, this is letter D. Number 38, so which is used to differentiate Proteus mirabilis from vulgaris. So both of these are gram-negative bacilli that are enteric, and they're known for being able to swarm. Okay, so they're also the aminase positive. Okay, so to distinguish the two, no, definitely we can perform your indole test. Number 38, letter C, indole test. Okay, so Proteus mirabilis is indole negative, while Proteus vulgaris is indole positive. Okay, so the positive result for the indole test is a red color, okay, a red ring or red color. Okay, so number 38, letter C. So to distinguish the two, so you can perform the indole test. Okay, 38, letter C. So nasal swabs, if you culture your nasal swab, no, so most likely what we can encounter from the nasal swab, okay, is your Staph aureus. Number 39, letter B, Staph aureus. No, so you have to take note, no, the reservoir of your Staph aureus is in the nares, or the nose. No, so majority of the Staph aureus are actually found there. Okay, so if you do a nasal swab, we can most likely isolate Staph aureus. Okay, Neisseria meningitidis can be isolated in CSF or even, okay, in your nasopharyngeal swabs. Okay, strep pneumo is an agent of pneumonia, but that may be also associated, that may be isolated in your sputum or even in your CSF as well. And Haemophilus influenzae can also be isolated in nasopharyngeal swabs and also in the CSF. Okay, but if it's for nasal swab, most likely you can isolate letter B, Staph aureus. Okay, 39, the letter B. Number 40, no, so the HASEC group. So the HASEC group is an acronym of related organisms. No, okay, so this includes your Haemophilus, the members of Haemophilus, except your Ducreyi and Influenzae. We have also your Actinobacillus, Cardiobacterium, Echinella, and Kingella. Okay, so what would be common among these HASEC members? No, they are all capnophilic. Okay, so they're all captophilic, meaning they prefer growing in increased amounts of carbon dioxide, 5 to 10%. They're all gram-negative bacilli. They're also non-motile. Okay, they're also non-motile, and they can be part of the normal flora. Okay, but all of them are able to cause opportunistic infections, particularly endocarditis. For number 40, this is letter B. So the particular type of endocarditis associated with the HASEC group is what we call as the SBE, the subacute, okay, subacute bacterial endocarditis. Okay, so for number 40, this is letter B. For number 41, which would be the correct sequence in the MPN method for water analysis? So the MPN method, or the multiple tube fermentation technique, the MPN or the multiple tube fermentation technique, or this is also known as the most probable number method, is considered as the gold standard, okay, gold standard method for water analysis. So the correct sequence in performing your water analysis using this method is, for number 41, it's letter A. So presumptive, followed by confirmed, and completed. Okay? So in performing water analysis using the MPN method, the total volume of water sample needed in this test is 100 ml. Okay? So number 41, this is letter A. Okay? So in the MPN method also, no, okay, we are detecting if the water sample is fecally contaminated, and our organism, no, representative organism, okay, to determine that there is fecal matter, no, in the water sample is Escherichia coli or E. coli. Okay? So that's the indicator bacteria. So meaning that if there is detection of E. coli in your water sample, it indicates it is fecally contaminated. Number 41, letter A. Number 42, no, which of the following is mismatch? So with regards to the microorganism and the characteristic feature, for number 42, this is letter B. Okay, so Ehrlichia and reticulic bodies. So this is mismatch because if you're dealing with Ehrlichia, you don't look for reticulic bodies, but rather look for morula. Morula bodies inside white blood cells. Okay, so Ehrlichia infections, these are vector-borne bacteria, no? Vector-borne disease, no? And in Ehrlichia, you look for the morula. Okay, so Streptobacillus multiformis, that's an agent of rat bite fever and Haverhill fever, so that's also associated with being the necklace bacillus. Okay, Actinomyces has that correct, that's correct. No, the molar tooth colonies, and Chromobacterium has that ammonium cyanide odor or almond-like odor, so which is also correct. So the one that is mismatched or incorrect here is number 42, letter B. Okay, number 42, letter B. For number 43, no, so an organism is identified as Listeria, and you want to perform additional testing. So what would you expect, no, if it's really Listeria for number 43? So you'd expect the presence of that umbrella growth pattern on SIM or on your semi-solid medium. Okay, umbrella-like pattern or growth or Christmas tree. So another description to that umbrella pattern on SIM is your Christmas tree. Okay, so Listeria is motile. It has the tumbling motility, no? So it's also able to grow at 25 degrees. Okay, so that's why the best answer here is 43, letter B. The other choices are all incorrect. Okay, since Listeria is motile and it is able to grow at 25. Number 43, letter B. 44. So which is able to support the growth of Legionella. Okay, so the growth of Legionella should contain which of the following additives? Legionella, this is a gram-negative bacilli that is highly fastidious. So meaning if it's highly fastidious, it will not grow on ordinary media such as your MacConkey agar. It will not grow in MacConkey. Legionella is also unable to grow in blood agar plate. Okay? So this particular group of bacteria are found in the environment, and they're usually isolated from air conditioning units as well as heating units. And it's usually associated with pneumonia, no? Legionnaire's disease, or that particular pneumonia, Legionnaire's disease among elderly males. Okay? So for the growth of Legionella, it needs your BCYE. BCYE. So BCYE, so that is your buffered charcoal yeast extract with cysteine. Okay, so the best answer here for number 44 is letter C. 44, letter C. Okay, number 44, letter C. Number 45, what's a rapid test that we can do to determine the presence of tubercle bacilli? If it's tubercle bacilli, that is your Mycobacterium tuberculosis, and it's also able to determine if the isolate is resistant to rifampin, which is the first line of drug for TB. No? So what is this rapid test? No? So this rapid test is actually a molecular test. A molecular test. No? So the best answer here for number 45 is letter C, Gene Expert. Okay, Gene Expert, this is a nucleic acid amplification test, or it's a type of molecular test, no, so that detects the specific nucleic acids of your TB. And alongside that, no, if it detects TB, it would also check for rifampicin resistance. Okay, for the Gene Expert, our specimen here is still sputum. Okay, for number 45, this is letter C. Okay, quantiferon can also be used for TB, but quantiferon is a IGRA test. So meaning if it's an IGRA test, this is a okay, so this is a test, no, okay, that would be detecting your interferon gamma. Okay, interferon gamma. An interferon gamma release assay. Okay, an interferon gamma release assay. No? So for the quantiferon test, our sample here is blood, heparinized blood. Heparinized blood. No? So to detect production of interferon gamma. Okay, so for number 45, this is letter C. Okay. For number 46, no, what is the most crucial step in gram staining? So this particular step, no, if it's not done properly, it may lead to a false gram positive or false gram negative result. So this is decolorization. So letter B. So the decolorization step using acetone alcohol. So that's the most crucial step. Number 46, letter B. So number 47, putting on of PPEs. So we call that as the donning. Okay, donning. 47, letter A. So donning. Now, if it's doffing, this involves removal of the personal protective equipment. Okay. For number 47, this is letter A. Okay. Number 48, which set of tests can differentiate Neisseria, most Neisseria, as well as Moraxella catarrhalis? So these two groups of organisms can be mistaken since they both appear gram-negative. No? They're gram-negative cocci in pairs. No? So for us to distinguish them, no? So the best test to differentiate them is letter B. Number 48, letter B. So we can perform DNase test, reaction on CTBA, so nitrate reduction and butyrate esterase. Okay, so DNase, okay, so DNase positive, that's Moraxella. Okay. Reaction on CTBA. So CTBA, that's used for carbohydrate fermentation. And we all know, no, Neisseria are fermenters of sugar. So you would expect a reaction on CTBA. Okay. Moraxella are non-fermenters, so you would have a negative result on CTBA. Okay. Nitrate reduction, so that's usually positive for Moraxella, and butyrate esterase, that's also usually positive for Moraxella as well. Okay. So 48 letter B. We cannot use the other choices since Neisseria and Moraxella are both catalase and oxidase positive. They share the same gram reaction. Okay. And they're also both non-motil. Okay. They're also both oxidase positive. Okay. Number 48, letter B. Number 48, this is letter B. Number 49, what is this exclusive pigment seen in Pseudomonas aeruginosa? So this particular pigment is only seen in Pseudo aeruginosa and not other species. Okay, so this is letter A, your pyocyanin. Okay, pyocyanin. So the blue pigment, no? So pyoverdine, okay, or this is your fluorescein. Okay, your fluorescein can also be seen in other species of Pseudomonas. No? So but pyocyanin can only be found in aeruginosa. Okay, violacine can be seen in Chromobacterium, Staphyloxanthine, that's in Staph aureus. Okay, prodigiosin, that's for Serratia. Okay, for number 49, this is letter A, pyocyanin. Okay. Number 50, so Trismus, lockjaw, and Rhesus sardonicus are all manifestations of which bacterial infection? So this is Clostridium tetani. Okay, this is Clostridium tetani. So your tuckhead bacillus. So tetanus caused by Clostridium tetani can be transmitted via puncture, no? Okay, puncture, okay, of contaminated materials, no? Or in some cases also we can also get tetanus after dog bites or animal bites. Okay, so you may also get this from animal bites, no? Aside from puncture. Okay. Number 50, letter A. Perfringens, no? For the other choices like perfringens that's associated with myonecrosis. Okay. Botulinum, that's food poisoning, canned foods, no? Difficile pseudomembranous colitis. Okay. Number 50, this is letter A. Number 51. So this capsule is composed of alginate. So this particular organism is known for being a non-fermenter. It's a non-fermenter. It's usually implicated in nosocomial infections. And this is usually seen among patients with cystic fibrosis. So patients with cystic fibrosis would usually have this organism that can lead to pneumonia. Okay? So this particular organism that has this capsule is your Pseudomonas aeruginosa. Number 51, letter C. Okay. Number 51, letter C. Number 52, which indicator is used to determine complete anaerobiasis? No? So meaning if oxygen is completely removed inside the anaerobic chamber, so definitely you need here that redox indicator. So you need a redox indicator to check if the oxygen is completely removed inside the chamber. So this is of course your methylene blue. Number 52, letter C. So methylene blue. Aside from methylene blue, we can also use resazurin. So that is another example of a redox indicator. For the other choices, no, we don't use acridine orange. That is a stain. The palladium, that would function as a catalyst to speed up the removal of oxygen. Okay. So number 52, this is letter C, methylene 2. So that would function as the redox indicator for anaerobic, for the anaerobic chamber. Number 52, this is letter C. Number 53, what is the expected KBC test result for Bacteroides fragilis? Okay. The KBC test, KBC stands for the three antibiotics. So that is your kanamycin. So we use here 1,000 micrograms of kanamycin. We have your vancomycin, 5 micrograms, and colistin, 10 micrograms. So the KBC test, now this is a test making use of these antibiotic discs, okay, antibiotic disc, as a preliminary test. It's a preliminary test for identifying common anaerobic bacteria. So we can use the KBC test to serve as an initial test to detect the presence of common anaerobic bacteria. So we can use the following antibiotic disc and we can check whether or not they are susceptible or resistant. Okay. So for it to be considered as susceptible, the cutoff is 10 millimeters. So a zone of inhibition of more than or equal to 10 millimeters would indicate it's resistant. If it's less than 10 millimeters, it's considered as resistant. No? If it's more than or equal to 10 millimeters, I mean that's susceptible. If it's less than 10 millimeters, it's resistant. Okay. So what would be the expected result? If it's Bacteroides fragilis, so letter A. So it would be resistant to the 3 antibiotic discs. Okay. So if it's only resistant to colistin, so this is typical of your Fusobacterium. Okay. So number 53, this is letter A. If it's resistant to the 3 antibiotics, that's Bacteroides fragilis. 53, letter A. 54, soft chancre is associated with, no? So the presence of soft chancre, this is your chancroid. So this is your genital ulcer disease. So this is a sexually transmitted infection wherein your patient would have these lesions or soft lesions that are painful, present in the genital area. And we associate this with your Haemophilus ducreyi. Haemophilus ducreyi is a highly fastidious member of Haemophilus, okay, that only requires X factor. It only requires X factor for growth. And this is associated with your chancroid. Okay? Haemophilus ducreyi is also considered as the smallest pathogenic bacilli. Number 54, letter A. Number 55, the first statement, swabs for viral culture should be made of cotton, dacron, or rayon on wood shafts. Okay, so since the swabs are placed in wood shafts, no, so we cannot use that. That's not preferred. No, preferably, okay, we cannot use wood shafts. No, preferably we want plastic shaft. No? So the first statement is incorrect. For the second statement, calcium alginate should not be used for viral culture since it may bind and inactivate the virus. So this is correct. Okay, we don't use calcium alginate for viral culture. It can be used for bacterial culture, but not for viral. Okay, so first statement is false. Second statement is true. This is letter B. Number 56, an agent of flesh-eating bacteria or necrotizing fasciitis. This is letter A, Strep pyogenes. Okay, so what do we know about Strep pyogenes? This is your beta hemolytic group A strep. Okay, beta hemolytic group A strep associated with scarlet fever, necrotizing fasciitis, and of course, this has that sequelae. So meaning there are complications, no? So due to Strep pyogenes infections, no, such as your acute glomerulonephritis as well as rheumatic fever. Okay, so key identifying tests for Strep pyogenes include they are taxo A susceptible, so meaning they are bacitracin susceptible. They're also PYR positive. Okay. Number 56, this is letter A, strep pyogenes. 57, so washer woman's hands, rice water stools are all associated with what particular disease? This is cholera. Number 57, letter A, cholera. So cholera is associated with your Vibrio cholerae. So particularly the O1 strains. No? Okay, the O1 strains of cholera are those that are pathogenic. Okay, so we can isolate your cholera, your Vibrio cholerae, using TCBS, very selective and differential for Vibrio. Okay, so number 57, letter A. 58, correct regarding Klebsiella pneumoniae. Okay, which of the following is correct regarding Klebsiella pneumoniae, except, so except, in other words, which is incorrect. So ano dito ang mali? Okay, so correct regarding Klebsiella pneumoniae, except, so for number 58, the best answer there is letter C. Okay, so it is indole negative. That's okay. So so for your Klebsiella pneumoniae, so this is letter C. Okay, so 58, this is letter C. No? So 58, letter C. So which is incorrect here, it's OMPG negative and motile. No? So Klebsiella pneumoniae, so that's lactose fermenter, so it's OMPG positive, not negative. No? And Kleb pneumo is a non-motile organism. So number 58, this is letter C. Number 59, so infectious jaundice or Wales disease are usually associated with infections caused by leptospira. Okay, number 59, letter B. So leptospirosis. So usually we get this by contact with contaminated urine, contaminated urine of rodents. No? So usually leptospirosis is suspected if there is history of wading in flood waters. No? So for number 59, that's letter B. So leptospira. Rickettsial organisms, usually we associate rickettsia for rashes. No? So the rashes, fever. No? Okay. So number 59, this is letter B. Number 60, which of the following characterize the group P non-enterococcus? Group P non-enterococcus, which is correct about your group P non-enterococcus. So letter B. They're able to grow in bile esculin media. On bile esculin media, you have that blackening. The colonies of this group of bacteria would appear black. Yep. So they're also able to cause that blackening. Okay. Since this is non-enterococcus, group P non-enterococcus, they would not grow on 6.5% sodium chloride. Okay. They're also not positive for PYR. Okay. Those that are positive, okay, are the group D enterococcus. Okay. So to summarize, no. So for the group D enterococcus and the group D non-enterococcus, okay, so if you do your bile esculin media, growth on bile esculin agar, okay, or BEA, bile esculin agar, both group D enterococcus and non-enterococcus would grow. No, you have that blackening. 6.5% sodium chloride, so that's enterococcus. It would grow in 6.5, but not the non-enterococcus. No? PYR test, group D enterococcus would be positive, while the non-enterococcus would be negative. Okay, number 60, this is letter D. 61, the Widal test. So this is a serologic test, no, for detection of antibodies. Okay, febrile antibodies or febrile agglutinins. So patients that are positive for the Widal test, no, okay, of course, are useful for typhoid fever. No? So for salmonella. Number 61, this is letter A, salmonella. Okay. Test for rickettsia, that's your whale felix. Okay, but if it's Widal, that's for salmonella. Number 61, letter A. Number 62, what is used to isolate Corynebacterium diphtheriae? The colonies appear gray to black. So this is letter A, your cysteine tellurite medium. Okay, 62, letter A, cysteine tellurite medium. Okay, so although we can also use Loeffler's serum slant for Corynebacterium, no, Loeffler's serum slant, that's useful for the metachromatic granules. If you're after the metachromatic granules, okay, we can use the Loeffler's. But if you want to isolate your Corynebacterium and you have that gray to black colonies, use your cysteine tellurite. Okay, 62, letter A. Number 63, what is the optimum growth of Campylobacter jejuni? Okay, so Campylobacter, these are microaerophilic organisms. So they're microaerophilic, you need reduced amount of oxygen. So around 5 to 10% oxygen. So the best answer here, okay, would be letter A. So reduced amount of oxygen, around 5 to 10%. So the closest answer among the choices is letter A. For number 64, now which the following are positive for the catalyst test. So this makes use of 3% hydrogen peroxide as your agent, and the positive result here is effervescence or bubbling. Okay, so which of the following is positive for catalyst? So definitely that's your Staphylococcus. Strep is catalyst negative. Okay, Bacillus, these are aerobic gram-positive bacilli, they're also positive. Okay, as well as Corynebacterium and Listeria. Erysipelotrix, that's catalyst negative. Okay, so the best answer here for number 64 is letter B. 64, letter B. 65, agent of trench fever. So trench fever, this is a vector-borne disease. Vector-borne disease, okay, for your trench fever, no? Okay, so that can cause a type of fever, no, that is relapsing. It's associated also with relapsing fever. So this is also known as the 5-day fever, meaning you would have a fever for almost 1 week and then it would disappear, or it would resolve and then it would go back again. So 5-day fever. So this is your Bartonella quintana. Okay, 65, letter B, Bartonella quintana. Okay, so from the root word quinta, meaning 5. Okay, for the other choices, no, your Bartonella bacilliformis, that's associated with Oroya fever. Bartonella henselae, that's the agent of cat scratch disease. And Orientia chuchugamushi is associated with your scrub typhus. Okay, so 65, this is Bartonella quintana, the agent of your trench fever. Okay, number 66, what's the function of your N-acetylcysteine? Okay, so used for digestion, decontamination of your sputum samples. So the NALC is primarily to liquefy the mucus. Okay, liquefy the mucus. 66, letter B. Okay, so always remember, for decontamination of sputum samples, the time needed to decontaminate the sample is 15 minutes. Okay, for decontamination, so 15 minutes. Number 67, gram-positive bacillus was isolated from a wound specimen and had the following results: double zone of beta hemolysis, lecithinase positive, lipase negative, indole negative. So since the wound specimen, double zone of hemolysis, or this is somewhat your bullseye. So this is Clostridium perfringens. So this is associated with gas gangrene or myonecrosis. 67, letter A. Number 68, which of the following is true regarding choleragen? Choleragen, that is the toxin responsible for the pathogenicity of Vibrio cholerae. So this is an enterotoxin, of course, that causes hypersecretion of water and electrolytes. No, so the choleragen is not invasive, so that's why letter B is not correct. So it will not cause, it will not allow penetration of the bowel mucosa since it's not invasive. Okay, and it's only produced by the toxigenic strains. Okay, so it does not really cause dehydration, but rather hypersecretion. So that's why the best answer here is letter A. 69. What is the TSI reaction of Shigella dysentery? So Shigella is just a glucose fermenter. It's negative for H2S, and it's also negative for gas production. So Shigella, you'd expect that if it only ferments sugar, no, you would only see the acid production in the bottom or in the butt, okay, in the butt part of the TSI tube. So in symbols, no, that is K over A. 69, letter A. K over A. Okay, number 70. What would be an acceptable specimen for detection of Bordetella pertussis, the agent of the whooping cough? So the whooping, so in Bordetella pertussis, no, causing whooping cough, no, it would usually affect the upper respiratory tract. So there would be a repetitive cough, no. Okay, so the preferred sample here, no, that we can submit for diagnosing Bordetella pertussis is nasopharyngeal aspirates. Okay, number 70, letter C. So since it affects primarily upper respiratory tract, so we don't collect sputum for that. Preferably, it's nasopharyngeal aspirates. Okay, float swabs are employed for strep pyogenes. Okay, nose swabs are useful for staph aureus. Okay, but if it's for whooping cough, it's nasopharyngeal aspirates. Number 70, letter C. For number 71, so bacitazine, that's your Taxo A, Camp Factor, PYR, and hipuric hydrolysis are most likely performed if you are dealing with what particular type of streptococci. No? So definitely for the following test, we can use this to differentiate your beta hemolytic strep. Number 71, letter B. Okay, your beta hemolytic strep. So for example, if you want to differentiate your strep pyogenes and strep agalactiae. Okay, so if it's bacitazine or Taxo A, strep pyogenes is susceptible, while agalactiae would be resistant. PYR test, pyogenes is positive, agalactiae is negative. Okay, CAMP test, positive is agalactiae, negative for pyogenes, and hipuric hydrolysis, positive for agalactiae, negative for pyogenes. Okay, number 71, letter B. Number 72, no, Morganellas, as opposed to Providentia, so these are gram-negative enterics. Okay, gram-negative enterics. So as opposed to Providentia, no, so they are letter A, citrate negative. Okay, so we can distinguish the two, no, using citrate utilization. So Providentia, you'd expect that they would be citrate positive, while Morganella are citrate negative. Okay, both of them are not fermenters of lactose. No, okay, so we cannot also differentiate Morganella and Providentia with the aminase since both of them are positive. Okay, so the best answer here is citrate to differentiate the two. Okay, 72, letter A. 73, anaerobic gram-positive. So take note, which of the following here is an anaerobic gram-positive bacilli. So, okay, so this is letter C, Propionibacterium. So Propionibacterium can be found as a normal flora of the skin. Okay, it can be found normally in the skin, and Propionibacterium is also a common contaminant of blood cultures. Okay. Peptostreptococcus is not the answer, although this is gram-positive, it's cocci. Okay, 73, letter C. 74, biosafety level 2 agents, no, are those that are common agents of infectious diseases. So BSL2. So this would include your enterics, okay, so HIV, okay, so the common pathogens that we encounter in the lab. And this is also the BSL level for the microbiology labs. Okay, so but if you're handling TB, no, and other airborne organisms such as Brucella, no, okay, it should be BSL3. For maximum containment facilities, that's BSL4. Okay, if it's BSL2, that's usually for the common agents of infectious diseases. 74, letter A. 75, first statement, streptolysine O is oxygen stable, produced by strep pyogenes. Okay, correct, it's produced by strep pyogenes, but this is not oxygen stable, it is oxygen labile. Okay, so although it's part of pyogenes, okay, this is still wrong since SLO is oxygen labile. So first statement is false. Streptolysine S responsible for subsurface hemolysis, that is wrong also because this is oxygen stable. So number 75, this is dog. Both are false. 75, dog. Number 76. So given the following results of an enteric organism, it's LDC, ODC, and EDH negative. It's triple decarboxylase negative. Okay, it's able to ferment glucose and lactose, no? And it's also oxidase negative. This particular enteric organism is usually associated with opportunistic infections in the hospital. They can also produce yellow pigmented colonies on your blood agar plate. Okay, they're usually also implicated in contaminated IV products or IV fluids, no? Milk products also, no? And of course, they're known for being LDC, ODC, and EDH negative. So this is definitely letter D, Enterobacter agglomerans, or now known as the Pantoea agglomerans. Okay, E. coli is not the answer since E. coli is LDC positive. Okay, Salmonella is also not the answer since it's a non-lactose fermenter. Okay, so number 76, letter D. 77, melioidosis. So this is associated with your Vietnam time bomb. So this is a potential bioterror agent caused by Burkholderia. No? Particularly what species of Burkholderia? That is Burkholderia pseudomallei. 77, letter D. So Burkholderia pseudomallei is a bioterror agent, and our culture media to isolate your pseudomallei is we can use your Ashdown medium. Okay, this is a potential bioterror agent. 77, letter D. 78, which mycobacterium? So that is associated with respiratory or pulmonary, gastrointestinal, and disseminated cases, and it's usually associated with AIDS patients. So this is particularly your Mycobacterium avium complex. Number 78, letter C. Number 79. So pieces of DNA that move from one genetic element to another. Okay, so they're able to jump. These are known also as your jumping genes. Okay, that can move from one element to another is your transposons. Number 79, letter A. So they're able to insert these transposons, or jumping genes are able to insert themselves, no, from one genetic element to another. Okay, So that is your transposons. Number 79, this is letter A. Number 80, so coccidioides imiti. So this is a dimorphic fungi associated with systemic mycosis. No, which is true regarding your coccidioides imitis. No? So coccidioides imitis, so histologically or in your tissues, they would have the presence of the spherules. Number 80, letter B. So the presence of the spherules containing the endospores. So this is actually the hallmark of coccidioides. So coccidioides is associated with your San Joaquin Valley fever. Okay. So for number 80, this is letter B. Okay, number 81, if you are using corneal agar and there's the presence of chlamydoconidia, particularly terminal chlamydoconidia, okay, this is typical of your Candida albicans. Okay, so what are the other features of Candida albicans? It's also germ tube positive. Okay, germ tube positive for Candida albicans. Number 81, this is letter C. Number 82, so first statement, Guarnieri bodies are found in patients with herpes. So this is incorrect. Guarnieri bodies, these are inclusions associated with smallpox. Okay, Guarnieri, not herpes. Okay, second statement, Councilman bodies are inclusions associated with smallpox. So this is wrong. If it's Councilman bodies, so this can be seen in yellow fever. No? So Councilman bodies, these are cytoplasmic inclusions that we can see in the liver of patients affected with yellow fever. So this is also wrong for number 82. So both statements are false for 82 dog. Number 83, which hair dermatophyte? So dermatophytes, these are fungi no, that would be targeting keratin-rich areas of the body. So it can affect the hair, skin, or nail. So for this question, no, it specifies that it affects the hair and it fluoresces yellow-green under the Woods lamp. No? So it's positive for the Woods lamp exam. Okay. So for the dermatophytes, we have your Microsporum, Epidermophyton, and Trichophyton. Okay, so these are the three dermatophytes, no? Okay, so it can affect the skin, the hair, okay, as well as the nails. Okay, skin, hair, and nails, no? So if it affects your skin and hair, but not the nails, that's Microsporum. Okay, if it affects the skin, not the hair and the nails, that's Epidermophyton. But if it affects the skin, hair, and nails, that is Trichophyton. No? So this is a hair dermatophyte, so we can already remove Epidermophyton as the possible answer. No? So if it fluoresces under Woods lamp, no, so definitely these are your Microsporum. So that's letter C, Microsporum autogini. So most Microsporum are positive for the Woods lamp, except Microsporum gypseum. Gypseum is negative for the Woods lamp exam. Okay, number 83, letter C. Microsporum albuini is an important agent of Tinea capitis, or ringworm of the scalp. Okay, so number 83, letter C. Number 81, the mode of transmission for most dimorphic fungi is, no, if it's dimorphic fungi, it can either become a yeast or a mold, depending on the temperature. So this is primarily transmitted via inhalation. For number 84, this is letter B. So inhalation of the spores. Okay, number 84, letter B. 85. So a stage of venereal syphilis caused by destruction, CNS disease, cardiovascular, eye disease, and granuloma-like lesions, so which we call as GUMAs. This is the tertiary syphilis, letter C. Okay, tertiary syphilis. No? So at this time, no, this can appear after many years from the time of infection, around 10 to 20 years. But in tertiary syphilis, the patient is already non-infectious. So this is your tertiary syphilis. Number 85, letter C. Number 86. No? So we have a patient diagnosed with HPV, okay, and she was not vaccinated with the HPV vaccine, or that's Gardasil vaccine. What serotypes of this virus? What serotypes of the virus does she need to be concerned about that? Okay, that would increase her risk of developing cervical cancer. So definitely, so the question is about the serotypes that are associated with cervical cancer, since the patient was not vaccinated against the virus. So for cervical cancer, definitely the important serotypes for that is number 86, letter C, 16 and 18. 16 and 18 are implicated in cervical cancers. Okay, number 87. So in viral culture, so of course, no, for us to demonstrate viral growth in cell cultures, we can look for the presence of the cytopathic effects, or the CPEs. So CPEs can be seen in the cell cultures. They can either be seen as in presence of inclusions, no, enlargement of the cells, no, okay, multinucleated cells, etc. For us to demonstrate this, we can use your phase contrast microscope, as well as the inverted microscope. Okay, so for us to assess if it is significant, okay, it should be at least 2+. Okay, so for 87, the question here is if the CPE grade is 60%, it is graded as 3+. So why 3+? No, if it's 3+, around 51 to 75% of the cells are infected. So this is the best answer, 87, letter C. 88. So the step in viral infection, wherein it involves attachment to the receptor. So this is adsorption. 88, letter T. Okay, number 89, what is an appropriate sample to diagnose tinea nodosa? Okay, so tinea nodosa, okay, this is also known as your piedra. So this is a superficial type of mycosis, superficial mycosis affecting the hair strand or hair shaft. Okay, so an appropriate sample of that, of course, is your hair shaft or hair specimen, the hair strand. So there are two types of piedra. It can either be black piedra or white piedra. Number 89, this is letter A. For number 90, no, so which of the following dermatophytes produce antler-like hyphal elements and very few spores? No? So definitely antler-like hyphal elements, or this is your fabric chandelier. So on your culture, so this is letter A, Trichophyton schonleini. Trichophyton schonleini is associated with tinea favosa. Tinea favosa is a severe type of tinea capitis that can lead to permanent alopecia. So one of the distinct features of that is the presence of this fabric chandelier or antler-like hyphal elements in culture. Number 90, this is letter A. Okay, number 91, which capsid is not seen or never seen in DNA viruses? Definitely, so that's letter B. So if it's a helical virus, that's always an RNA virus. So we don't encounter helical viruses or DNA. That's always an RNA virus. 91, letter B. 92, Owl's eye inclusions. So these are inclusions found in the nucleus, are very typical of your cytomegalovirus. Number 92, letter D. Okay. If it's Epstein-Barr, you look for the Downy cells. The Downy cells, no? Adenoviruses, you have the grape-like clusters. Rabies, you have the Negri bodies. Okay. So number 92, this is letter D. Number 93, so QC, quality control for hair beating test. So in this case, you have to look for a positive and a negative control. No? So one should be positive, the other should be negative for the hair beating test. So definitely this is letter A. Hair beating test, you have rubrum and mentagrophytes. You'd expect mentagrophytes to be positive for the hair beating test. Rubrum would be negative for hair beating. Number 93, letter A. For number 94, no, the basic functional unit of molds, of course, that's your hyphal elements or hypha. 94, letter A. Mycelium is the group of hypha or a aggregate of hypha. So the basic functional unit is hypha, letter A. 95. What is the all-purpose medium for the isolation of fungi? So that's definitely your SDA or Sabouraud's dextrose agar. 95, letter A. 96, spaghetti and meatballs on direct QH, of course, are seen in cases of tinea versicolor. 96, letter A. So tinea versicolor is caused by Malassezia furfur. 96, letter A. 97, hallmark of congenital rubella is the blueberry muffin. 97, letter A. 98, so predominantly associated with respiratory epidemics of keratoconjunctivitis or sore eyes. We usually associate this with a DNA virus, and that is your adenovirus. 98, letter A. Norwalk, that's usually diarrhea among adults. Rotavirus, diarrhea among infants. Okay. Molluscum contagiosum virus, now we have there those lesions on the skin. 98, letter A. 99, letter B, I mean. For 98, this is letter B, your adenovirus. Number 99, the most common viral syndrome of pericarditis and myocarditis is usually caused by your Coxsackie. 99, letter C. And 100, which of the following is a major biohazard to laboratory personnel. So you have to handle this in a BSL-3 laboratory. So that is your coccidioides imitis. Okay. So for number 100, that's coccidioides imitis.